Well, good morning, everybody. My name's Ryan Murphy. I'm with the University of Minnesota. Welcome to the MnSTAC Monthly Forum. This is a monthly forum series that we put on in collaboration between the Urban Forestry Outreach and Research Lab and the Minnesota Shade Tree Advisory Committee. Just a quick reminder that this session is being recorded and it will be made available after the session on our YouTube page. Just post that link in here. And if you haven't already, please do sign up for the monthly or the weekly MnSTAC newsletter. You can do so here at mnstac.org. Send out a weekly email and about all things urban and community forestry. So today our presenter is Brett Arenz. Brett is a teaching associate professor and also the director of the Plant Disease Clinic in the Department of Plant Pathology at the University of Minnesota. In this talk, Brett will cover a variety of tree and shrub diseases that commonly are being submitted to the Plant Disease Clinic, as well as some emerging pathogens to be on the lookout for. Thanks everybody for being here. I know it's a bright day and I'm sure there's plenty to do outside, but it's great to have you here. And Brett, thank you for being our presenter, and we look forward to your presentation.
All right. Thank you very much, Ryan. It's always a pleasure to talk to this group. I know I've done some talks in the last few years in the sort of outdoor setting and field house. I think this is the first Zoom talk I've given to this group. So, as Ryan mentioned, I wanted to give you a kind of a quick overview of our last year in the clinic, what that kind of was like. Just a little bit of reference here. This is everybody I know had probably, I'm guessing, had different hobbies you picked up during the pandemic when you were stuck at home. Mine was chainsaw carving because I had an oak tree next to my house that was dying from armillaria root rot. So I had to get it cut down. And, I had them leave kind of about 6 feet of stump because I always figured I wanted to do something with it. I wasn't actually able to find a chainsaw artist that wanted to come in and work with it. So eventually, after 4 or 5 years, I decided to try it myself with a relatively simple design. And it turned out okay, and I didn't lose any limbs in the process. So let's get started here. Just kind of another quick overview. So I just wanted to quickly mention, kind of go over what the Plant Disease Clinic is, or as I'll kind of call it, the PDC, especially for folks that maybe have not used the clinic before, or maybe are newer to MnSTAC. And then kind of going into a review of 2021 season, so last year. So things that kind of stood out in 2021 that made it a little bit different from other years. But also I'll be covering some of our more typical tree submissions as well. And then finally, I'm kind of wrapping up talking about tips for sample submission. If you are interested in submitting samples to the clinic, or if you're trying to give recommendations to people that are interested. And it sounds like we'll have plenty of time for questions as well. If you have any, feel free to put them in the chat, or you can save them for the end as well. Okay, so first of all, what is the Plant Disease Clinic? It is a lab, a fee-for-service lab, that is on the St. Paul campus. And we provide diagnostic assessment of plant health problems. Okay, so I say plant health problems because, it's not always disease that we find on these plants. And I'll get into that in a second here. But just as kind of a point of context, we had just over 2,500 diagnoses that we made in last year. And it was actually over 170 different plant host species. So we see a really wide variety of types of plants in the clinic. Obviously, samples submitted by farmers, a lot of corn and soybean, different type of vegetable crops, a lot of ornamentals will come into the clinic. There's quite a few big producers in the metro area that will submit things as they're ramping up production. Also homeowners as well, some of their favorite ornamental species, but of course, trees, trees and shrubs submitted either by homeowners or by tree care companies themselves is a really big source of samples for us. But as I mentioned before, this plant health problems, why do I say that instead of disease? In any given year, quite a bit, about 30%, I should say, approximately, are actually no pathogen found as a diagnosis. And usually these are things like nutrient issues, either deficiencies or even toxicities in some cases, insect problems, environmental damage, things like cold damage, things like that, herbicide, chemical damage, herbicide drift can be fairly common certain times of year. So in addition, we also provide kind of a commercial research support on a contract basis. So these are maybe, for example, different companies that are trying to develop perhaps new products for pest pathogen control, right? But a lot of these companies actually don't have their own laboratories to do some of the testing, especially testing on microbiological types of things. So they need a lab that can actually do some sort of in vitro test of a product they're actually developing. So we do that by arrangement as well. So most of the funding we get actually comes from our clients in the form of our fees that keeps our lights on and keeps us running day to day. Okay, so just as kind of a quick rundown of 2021. So we had just over or almost 2,300 samples. Okay, so in some cases you get multiple diagnoses on a single sample, so that's why you get more diagnoses than you get samples. Most common sample types, I just have the top 4 here: pine, oak, garlic, spruce. Sometimes we joke that we're going to change our name to the Pine, Oak, Garlic, Spruce Lab instead of the Plant Disease Clinic, because these are such common types of samples for us. And pine, for example, you might be wondering why we had so many pine, almost half of our samples. It was actually a large survey that we did for Diplodia on red pine seedlings. Okay, so I'll talk about this disease in a bit here and why it's such a big deal, sort of towards the end of the talk here. And oak obviously is a big one. I'll spend a lot of time talking about that. Garlic, you're probably wondering what the heck is going on there. Well, as it turns out, there's actually quite a big disease problem in garlic called the garlic bloat nematode. It's a microscopic worm, basically. So you can't see it without magnification. And it's not like, it's not even closely related to an earthworm. It's basically a microscopic roundworm that can be devastating to garlic production. And when farmers, these producers of garlic, who oftentimes invest, quite heavily into garlic production, thousands of dollars to produce garlic on a few acres. Once they get it into their field, it actually prevents them from growing garlic there for several years, okay? Because there's essentially no resistance in garlic to this pest. So the garlic industry is trying to be very proactive in trying to prevent the spread of this nematode. And it basically spreads by people trading garlic bulbs, or someone buying garlic seed in the form of bulbs to plant in their farm. And in a low level, they don't know that there's infection there, and so that's how it gets introduced to different farms, basically. So most of this is proactive testing to try to screen bulbs that people have brought in to make sure the garlic bloat nematode isn't on there. And then also spruce, almost 100 spruce samples as well. We've assisted with various surveys last year, this Diplodia survey I was talking about. Also tomato brown rugose fruit virus, sometimes just called Tom Brown because of its acronym. This is a fairly important virus affecting tomato that's kind of been spreading sort of throughout the U.S. in the last few years. And fortunately, they've been pretty successful at eradicating it when it does pop up, okay? But why it is especially important is because it basically overcomes the resistance that they've been successful in introducing in tomato to the tobacco mosaic virus. Okay, so it's a very similar virus, but it has the ability to overcome the resistance. That's why it's important. Alternaria species and crucifers, these are things like Brussels sprouts, kale, things like that. Alternaria is a big cause of leaf spotting, and trying to determine what species are actually the most important when they emerge was kind of the objective of that study. Corn tar spot is kind of an emerging disease on corn, just sort of reported in Minnesota in the last couple of years. It's been kind of spreading throughout the US. And obviously corn, it's hugely important in Minnesota, along with soybeans, the top crop essentially. And so, the impact on yield of corn tar spot can actually be pretty significant in certain years. So trying to track how it's been spreading through the state is the objective there. And then also elm yellows, which is actually a phytoplasma disease that can affect elm. Obviously, it's called elm yellows because it causes the leaves to turn yellow and tree to get kind of stunted and unhealthy. So still some dispute as to if Minnesota actually has had elm yellows before or currently does. It was reported in the 70s, but it was only really based on symptoms, I believe, that this was reported. And obviously, well, maybe not obviously, but you can actually only confirm it through the use of molecular means, through DNA testing. And in the 1970s, they simply didn't have that available as an option. So we're trying to determine if elm yellows actually is present in Minnesota or not. Haven't found it yet in these surveys. Okay. So I've kind of distilled here the types of samples that we get and really removed everything that wasn't a tree, okay? So really concentrating on the trees here, obviously, from MnSTAC, and essentially ordered it by count. So the most common stuff at the top, least common at the bottom, and I've only, selected the top 18 or so here. All right, and then I've kind of organized them by host group, and then you can see the pest common name, then followed by the pest scientific name, and the number of times we diagnosed it in 2021. All right, so just want to quickly point out, first of all, that you can see there's a number of abiotic disorders on here in spruce, pine, oak, also even apple, cedar, and juniper here. So this is really not too unusual in any kind of given year, but what was sort of interesting about last year, I'm sure a lot of people remember this, is that it was exceptionally hot and dry, especially the first part of the year. This was the sort of drought report generated in August last year by the well, I'll say just the official drought report here by the USDA. And August was really sort of the height of the drought period. After that, we actually did get some rain in Minnesota and did get a little bit of recovery, at least in the fall, although I would say probably trees still went into the winter fairly stressed overall. But as you can see, as of August, most of the state was either in extreme drought in red here, or this dark orange, which was severe drought, right? I'm sure everybody kind of remembered that period. Very, very different spring last year than what we've seen so far. Okay, so obviously this was really stressful for a lot of trees. The more classic case that we would see in the clinic is we would say, well, someone would call us up or send us a picture and say, my tree is totally brown, it's all wilted, I want to get it checked for disease. And, we would ask them, say, okay, well, what's the history of this tree? In a lot of cases, they say, well, it was planted a month ago or 2 months ago. And they say, I've been watering it, but, in these cases, transplant shock is a really significant, cause of health problems, especially for some of these larger trees that sometimes people buy. It takes a lot of time in multiple years in a lot of cases for some of these trees to actually get established. And very difficult to do so when you're in the midst of a very hot and dry season. Okay. All right, so with that being said, let's talk about one of the more common types of trees we get. Obviously, oak. We saw, as I mentioned before, over 300 different oak samples submitted. And by and large, most of these samples were submitted because they wanted them tested for oak wilt. And we did find that we had positive confirmation 61 times last year. So everybody, I'm guessing on this call has probably heard of oak wilt. I'll just kind of give you a kind of a quick rundown and review. So oak wilt is a fungal vascular disease. It affects all oaks, but of course there is quite a bit of difference in severity. You know, the red oaks, things like, you know, red oaks and northern pin oaks, things like that, things with the pointed lobes, right? These can be affected very severely and they can die within a couple of months of being infected. Whereas whites and burrs, the other main types of oaks in Minnesota with more kind of rounded lobes, these can be, you know, affected and can look, you know, pretty bad from oak wilt, but they don't tend to die nearly as quickly as the red oaks do. And also you can even in some cases, if it's early enough, you can potentially treat them, you know, with some of these injectable fungicides. Whereas in red oaks, really, it's not viewed as useful. Once the tree is already infected, it will die, and a fungicide's not going to stop that. So this is the current map of oak wilt as put out by the DNR last year. You can see the sort of brown kind of shaded areas. That's where it's been reported in Minnesota so far. So obviously the metro area is right in the middle of that. Huge problem there, but then also kind of sneaking up the, you know, the Mississippi River, you know, past St. Cloud, getting closer to Brainerd, and then also kind of the 35E kind of corridor up towards Duluth, right? So the DNR is especially interested in areas outside the light brown areas to see if oak wilt has been found, and potentially, you know, they will even try to eradicate it if it actually has been found outside of this shaded area to try to stop the spread of this disease. It is considered an invasive disease. It has been here for quite a while, though, you know, ever since the, well, at least the 60s, it's been found in Minnesota. So it's just actually kind of interesting. It has not potentially spread as maybe as much as it could have, you know, compared to some other invasive diseases that we've seen in the past. But the nice thing about that, obviously, is that it does have the potential to, you know, try to prevent its spread because of that, if you can find it early enough. Okay, so one of the big things about oak wilt, of course, is that when you think of long distance movement of this pathogen, it's mainly spread by these bark beetles. Okay, and bark beetles are attracted to fresh wounds. There's research that's been shown that they can actually locate you know, cuts on oaks within a few minutes. And if they have the oak wilt spores in them and they start tunneling in and feeding on the oak tissue, that's how trees can get infected. And it's especially important because of this to check the oak wilt risk status before pruning healthy oaks. Okay, that's one of the #1 things we tell people if they're trying to prevent their trees from getting infected with oak wilt. Make sure you're not pruning during the high risk period. And the high risk period is really when these beetles are active. Okay, typically it goes from about April to July or so. And obviously we're in the high risk period now. It changes a little bit by year. You know, I would say probably it got started a little bit later this year because we did have a relatively cold spring, certainly compared to last year. But you know, that's kind of the rule of thumb. April through July, definitely a high risk period. You can actually check the extension web page here to see if we're in the risk period or not, right? And so during this high risk period, definitely do not prune. If you have to prune, maybe some sort of emergency cut or something like that. Or maybe you're trying to, you know, actually take a sample for oak wilt testing, you know, collect some of these branches. It's recommended that you treat the branch stubs with some sort of shellac immediately, basically to prevent the bark beetles from being attracted to these fresh wounds. And, you know, I should also mention that it can spread by root grafts as well. So it doesn't necessarily just need the bark beetles, but of course the root graft spread is going to be only to adjacent trees. So that's not really how it moves, you know, throughout Minnesota. It's really via these bark beetles. Okay. So just to kind of give you a rundown of what we found last year with this oak wilt testing. So I'm just going to talk about this table a little bit. So I've kind of broken it down by oak wilt type, either white, bur, or red oaks. And then you can see the number of samples that we actually received in the clinic last year. And then I have the percent of each of those types of oaks that were actually positive for oak wilt. Okay. And then there's also, two-line chestnut borer. Some of you may have spotted that on the top of the table. Basically, while we're doing this oak wilt testing on samples, we also check it for other pests and pathogens. The two-line chestnut borer is one of the common things that we see on some of these samples. Then also Bur Oak Blight is another fungal pathogen I'll talk about in a second. And then we have this sort of other category, which can include a lot of different things like oak anthracnose, tuberculous leaf spot, Botrytis twig canker, or even some sort of other insect problem, maybe. Okay, so just quickly to point out here, as you can see, a lot of the oaks that we receive actually don't test positive for oak wilt, even though that's the main reason they've been sent to us, okay? Because obviously oak wilt being a devastating disease, individual oaks, can add thousands of dollars to your property values. If you think of sort of a really large charismatic tree in your front yard. But if they're dying or they need to be removed, that can also cost thousands of dollars. So knowing if a tree has oak wilt or not can be extremely important for homeowners. Okay, so that's why we receive a lot of oaks for testing. But as you can see, in a lot of cases, especially the whites and burrs, trees that are suspicious for oak wilt and people want to test them, as it turns out, in a lot of cases, they don't have oak wilt. So why are they being submitted when they don't have oak wilt? Well, they can maybe have other things that are actually what we call look-alike conditions. Okay, so two-line chestnut borer certainly is in that category, can cause very similar symptoms to oak wilt. And I believe it's especially the reason we get a lot of whites and burrs in the clinic that actually don't have oak wilt, probably because they have this, this insect issue instead. Bur Oak Blight, as I'll mention, I'll talk a little bit more about that in a second. And then this other category. So in this other category, oftentimes we have a lot of the other foliar fungal pathogens like anthracnose and tuberculia. Normally this is higher. This percentage is higher on sort of normal years. And that's because typically we have more precipitation than we did last year. And because that's a huge factor for a lot of these other types of pathogens, okay? So that was kind of an interesting thing about last year is this other category was fairly low. And, kind of when we were in the midst of spring, kind of about the same time last year as we are in now, maybe about May, it had been really hot and dry. It still wasn't the primary period of time where we get oak wilt samples because that's usually about the middle of the summer when the symptoms start to be expressed. We were asking ourselves, gosh, maybe this is the year where we don't get a lot of oak wilt samples because it's not good conditions for some of these lookalike diseases. So maybe people won't be as concerned about their oaks because they won't have as many sort of, fungal foliar issues. But as it turned out, we still had a ton of oaks. I think what was happening is the drought stress was causing kind of similar symptoms on some of these oak trees. And, really that's sort of the take home point for me is that it doesn't really matter the type of year, wet years, dry years, we still get a lot of oaks in the clinic because there are different factors that cause some of these kind of lookalike conditions. Okay. All right, so just kind of to get into some of these lookalikes, two-line chestnut borer was actually the most common diagnoses we had on trees last year. So the symptoms on the trees can be, extremely similar to what you would expect with oak wilt, sort of browning of leaves in the upper canopy, typically kind of starting out there and kind of moving its way down throughout the years. And a lot of times, when we get these branches that are submitted for oak wilt testing, we just see the tunnels, okay, in the actual branch, and we don't necessarily see these larvae, right? These distinctive two-line chestnut larvae. So in a lot of cases, these diagnoses are suspected two-line chestnut borer, But it should be also said that there's other types of insects that can cause, sort of significant damage to oaks, such as like a flathead apple borer, and some of these bark beetles actually can cause kind of similar symptoms. But it does seem as though this two-line chestnut borer seems to be the most common culprit. It is actually considered a native insect to Minnesota, so it's not invasive. And mainly, it's thought that stressed trees are really what's going to be the most susceptible in this case. Healthy trees seem to have a pretty good ability to resist significant two-line chestnut borer damage, okay? So really, as far as management recommendations go, trying to reduce stress on oak trees seems to be the most important thing. What they actually do is the larvae will kind of feed on the vascular tissue underneath the bark, and then they will actually emerge as adults. They have a winged form as adults that will fly around and kind of feed on leaves on the oak tree for a couple months in the summer, and then they lay the eggs again on branches, and then the larvae, in fact, throughout the year. It takes usually about a year for them to complete that life cycle, in some cases even 2 years. So that's why, like pruning, therapeutic pruning of oaks, getting rid of these dead and dying branches, which will usually host a lot of the two-line chestnut borers, is considered a good way of kind of reducing inoculum, or I would say pest presence in this case, on individual trees. All right, so as I mentioned, very hard to tell outside of the tree, even for very experienced tree care professionals, it can be very difficult for them to distinguish two-line chestnut borer versus oak wilt. And that's why they send us a lot of the samples so we can actually do culturing tests and DNA tests for oak wilt. But if you do get the branches down and you can look under the bark, there are some really interesting things you can look for under the bark itself. So oak wilt, just looking here on the right, the discoloration you see on the wood is typically kind of gray and kind of diffuse, kind of streaky in this way. As you can see, it kind of gradually blends from the healthy to the kind of discolored wood. Whereas on the left, this is sort of a classic kind of two-line chestnut borer gallery. You can see that they're very sharp. Okay, it goes from dark to kind of healthy wood in a sharper line, and it's also very meandering. Okay, so it's not just kind of streaky down the direction of the branch. It can make turns, it can go in different directions, right? It can be sort of maze-like in appearance. It can get tricky sometimes when you have both of them on a branch, right? Which can be possible. So just because you find insect galleries doesn't mean the tree doesn't necessarily have oak wilt, right? But it can be a really useful thing to look for. Okay, let's get into some of these other look-alike diseases. So if you remember, on the table I had showed you, this was on 15% of the bur oaks received last year. It's something called Bur Oak Blight, also in a lot of cases just called BOB because of that acronym. It's a fungal pathogen, Tabacchia iowensis. Kind of an interesting one. It's only really been kind of reported since the 1990s, and really only seemed to become significant, I would say, in the last 10 to 15 years, right? We're not exactly sure why that is, okay? It's, as I mentioned, it's a foliar pathogen. It's definitely more associated with the veins. And I'll show a comparison picture in a second here than actual oak wilt. You don't typically get significant symptoms until the later part of the season, okay? Which actually, can be similar to oak wilt in that case. And then another kind of interesting symptom is that the leaves that are affected by Bur Oak Blight oftentimes will stay attached to the tree over the winter, whereas normally bur oaks will shed their leaves, before the winter. This is a really interesting one. We've talked a lot about it in Minnesota in the last 10 years. There's been a little bit of maybe disagreement, controversy, I should say, about how significant it is. And it can be actually pretty hard to predict the course of this disease on individual trees, okay? In some cases, it seems to get worse and worse on a tree over several years. In other cases, you can have a tree that's really severely affected, a lot of brown leaves on it, and then the next year, the leaves are green, right? And the new leaves, I should say, are green, and it has a pretty good, a season. So in some cases, people feel as though maybe it's not as significant a health problem, perhaps, as we initially thought. But it's something we're still trying to pay attention to and tracking to see if it becomes worse or what happens with it. Just kind of a comparison show here with Oak Wilt versus Bur Oak Blight. So obviously with Oak Wilt, it's more of a kind of a marginal necrosis that you start to see the initial symptoms before the leaves kind of turn totally brown. You can see kind of red oak here and then white oak, and kind of working its way down to the lower part. With Bur Oak Blight, as hopefully is obvious, it's only on Bur Oaks for one thing. And also it's much more associated with the individual veins. So it can be vein delimited in some cases, as you can see kind of the wedge shape at the end here. But then even the veins themselves can actually start to become necrotic. And that seems to be where it kind of starts in other cases. Okay, so this is called Tabacia on oak, right? And I'm kind of clumping two different diseases here. Bur Oak Blight is a Tabacia, okay, Tabacia ioensis. And there's another disease, which is another Tabacia, Tabacia dryina, which causes Tabacia leaf spot. I'll say a little bit more about that in a second. But just wanted to kind of show some multiple year data we've had on this genus. And this is just based on samples that have come into the clinic, right? And I should clarify that this is not a scientific survey by any means. We're just tracking what people have sent to us. Going back from 2016 up till last year, okay? So the dark blue is the Bur Oak Blight, and the light blue is Tabacia leaf spot. You can see for the individual years, basically just the number of samples we received. Well, as you can see in one case, 2018 in particular here, we were really surprised because we had a very low amount of Bur Oak Blight that came into the clinic. And this was following several years where it was a very common sample type, and there was a lot of concern for it. But in 2018, seemed to be not a problem, okay? And the best sort of hypothesis we've come up with is that there was a relatively dry period in May, maybe for a couple of weeks in May, when the leaves were emerging. And that seems to be the most prominent time these leaves, the most important time, I should say, for infection from the spores. That's when the leaves are most vulnerable to being infected by the spores. If they get past that period, without being infected, they have fairly good resistance to the fungal pathogen here. So again, this was not a tested experiment by any means. This is just the best sort of guess we've come up with in this case. Okay, so we're kind of asking ourselves, well, gosh, in 2018, relatively low amount of Bur Oak Blight, maybe Bur Oak Blight's not going to be such a problem in the future. Let's see what happens. Well, wouldn't you know it, 2019, it definitely seemed to kind of bounce back from where it was, especially the Tabacia leaf spots, a lot of it on samples. And if you recall, in 2019, we actually had record precipitation in Minnesota, really wet year, especially during that sort of period of leaf emergence. So that seems to be a really key sort of piece for Bur Oak Blight. And then just to kind of quickly point out, of course, 2021, relatively low amount of Bur Oak Blight, also Tabacia leaf spot. And of course, it was a really hot and dry year as well. So I'm guessing that was probably the reason for that. So this year, it's been a relatively normal year so far, pretty decent precipitation, obviously, in the last month or so. So I'm guessing we're going to see Bur Oak Blight again this year at relatively kind of normal levels that we've seen the last 10, 15 years. Okay, so as I mentioned here, this Tabacia leaf spot, this is the other species, as you can see, looks a bit different than Bur Oak Blight. Another important thing is that it's not just on Bur Oaks. You can find it also on white oaks, and then especially on red oaks, it seems to be more common on. Again, like Bur Oak Blight, though, it is relatively late season, so it's not really until in the later part of the summer that you'll start to see more kind of significant necrosis. Not as associated with the veins. In some cases, you can see some kind of coalescing areas here, but it has a more kind of distinctive leaf spot, which I guess is more typical for a lot of foliar fungal pathogens people are familiar with. It can cause early defoliation if it's a relatively significant year. So trees that are severely infected can drop their leaves, maybe even put out, in some cases, they can put out more leaves. But because it's relatively late in the year, it's not considered to be a big health problem because the trees have done most of their photosynthesis kind of before this happens. And again, just to reiterate, this was not a widespread problem last year. Okay, so this is actually a great question here in the chat. What's the difference between oak blight and oak anthracnose? That leads me to the next slide, which is on oak anthracnose. So oak anthracnose, kind of similar type of disease to tubercular leaf spot in that it is a fungal disease and it is foliar. So basically just affecting the leaves and possibly the twigs of the oak trees, doesn't get into the vascular tissue. This, on the other hand, tends to be more severe on white oak, and it's actually an earlier season disease. So cool, wet springs tend to favor it. And, you know, if you think of what we've kind of gone through the last month or so, I would not be surprised to be seeing some oak anthracnose in the next month. Oak anthracnose, as far as symptoms go, you can get leaf spots. You can get larger areas of necrosis when these leaf spots kind of come together. Because it is infecting the leaf when it's growing, you can get areas of necrosis in the leaf, and then the leaf will continue to grow. And because of that, it tends to get kind of distorted and it looks like kind of twisted and like it's not grown right. Oftentimes it gets mistaken for herbicide drift because there's a lot of herbicides that actually cause kind of a similar type of leaf malformation as well. This can cause early leaf drop. And because it's an earlier season disease and that early leaf drop is more of a problem for trees, it can be a bit more significant for the overall health of the tree. Certainly, you know, not as significant as oak wilt by any means, but I would say a bit more significant than tubercular leaf spot. But, you know, trees can actually put out more leaves after this happens. So they can recover from it quite well in a lot of cases. But again, as I mentioned before, not a widespread problem in 2021, but I would expect it to be, again, more of an issue this year based on what we've seen so far. Okay, so thanks for hanging with. That was most of the oak wilt stuff I wanted to talk about. Obviously, the bulk of the talk. Just I wanted to mention a few more diseases that are relatively common diseases for us in the clinic. Third most common here on spruce is the spruce needlecast, in this case caused by Stigmina lautii. So these spruce needlecast diseases oftentimes have very characteristic types of symptoms in that they are typically infecting the older leaf, or excuse me, they're causing the necrosis in the older needles first because it requires a couple of years, a year or two at least, for them to infect, get into the needles, and actually cause problems. That's why you start to see browning on the older needles first. And the overall symptoms on the tree, oftentimes it's the lower branches and it's the inner parts of the branches that are infected. And oftentimes at the end of that, you just have a few needles kind of on the end, which is like the newest growth, and eventually that branch will die. And oftentimes leads to these kind of lollipop looking trees. Okay, so the Stigmina needlecast is definitely the most common that we have been seeing in the last few years. This is a fungal pathogen of spruce, obviously. And I mentioned, okay, a little bit already about the older interior needles. It tends to affect it first. Obviously, this is exactly the opposite type of symptom as you would expect with drought stress, which is mainly going to be the ends of the branches and the tips of the needles that are going to be browning first. So it's fairly easy to differentiate from that. And also winter burn has kind of a similar kind of symptomology as well, because really winter burn basically is drought stress. You know, think of what's actually happening. And what's interesting about this one is it's actually replaced a different fungal pathogen, a very similar fungal pathogen called Rhizosphaera needlecast that causes very similar symptoms. And it seems to be since about 2002 when it was kind of first reported in this area, that it's really, at least as far as what's coming into the clinic, has become much more common than Rhizosphaera. Okay, Unfortunately, I don't think there's any one group that's really studying this, you know, in a sort of really definitive way. So there's a lot of information we still don't have about it, how it's actually different than Rhizosphaera. As far as control goes, there's a lot of variability between spruce trees and how susceptible and resistant they are. Colorado blue is considered the most susceptible. Maybe Norway spruce is perhaps the most resistant. And then all the other spruces are kind of in between those two. Because it is a fungal pathogen, increasing air movement is considered to be really important. So making sure the spruce trees aren't crowding each other. You know, a lot of times people will plant little spruce trees too close together, and then they're fine for a long time, and they start to grow into each other and cause stress problems. People oftentimes have irrigation systems that will cover the branches of their spruce trees. That is an ideal situation for this fungal pathogen. You can use fungicides, but it's really only to protect the new growth. Okay, once a needle is infected, this is kind of what it looks like when the fungus is emerging from the stomata, it can't be cured of the pathogen. Okay, so basically these spores will get out into the air and they will get onto the new growth, and that's how it kind of repeats in its disease cycle. But if you can coat the new growth with some of these fungicides, you know, maybe when they're about halfway grown and then kind of repeating the treatment when they're fully grown, it is seen as a way of protecting that new growth from infection. Okay, but it's obviously something you're going to have to repeat, you know, and be very diligent about. Okay, I'm not going to talk about all of these. Obviously, we don't have time, but I do want to kind of talk about some that are really common types of samples for us in the clinic. And moving down to pine here. Didn't have a lot of this last year, which was a little bit surprising, but Diplodia tip blight is a really common type of sample for us. Most pines in Minnesota are susceptible, okay, especially the non-native pines, you know, things like the Austrian Scotch pine, you know, Yugo pine, but also red pine can be pretty susceptible. Okay, but and also things like jack pine. So really it's the 5-needle pine like white pine that's considered to be very resistant to it. Okay, and we very rarely see that on white pine. Okay, so very common. It actually has a very distinctive symptom when a tree is being affected by Diplodia tip blight. It's going to be the newest growth that is affected first, so kind of the opposite of the Needlecast on spruce. And oftentimes what you'll see is a branch that's green but has a brown end. And what happens is that actually the pathogen will kill the end of the branch as the needles are growing. So oftentimes these brown needles at the end are like short because they haven't finished growing. Okay, and so that's a really distinctive way of being able to tell this apart, maybe from other types of things like, you know, simple drought stress. And then these stress trees are really going to be the most vulnerable. So a tree stressed by drought, for example, can be much more prone to Diplodia tip blight than perhaps some other healthy trees. It is a latent pathogen, which means that a tree can become infected and have no symptoms at all until it becomes stressed, and then the pathogen becomes active and it kind of accelerates that mortality that you start to see. So really, this was why we were doing this survey that I mentioned right at the start of this talk, to try to basically screen a bunch of red pine seedlings to see if they had latent infection from Diplodia, okay, or if they were indeed not infected. Okay, moving down to another one that actually is an interesting one that has become fairly common in the last couple of years, and that's actually on lilac. And this is lilac leaf blight. So this is another fungal pathogen. And it's largely been reported as Pseudocercospora. Some other groups like to use the name Septoria. Basically, as far as I can tell, this refers to the same pathogen. Okay, it's sort of I don't need to go on to it too much, but with fungal taxonomy, you oftentimes run into these situations where there's some disagreement about what name to actually give things. And so this is a case, that's happening here. So Pseudocercospora, Septoria, whatever you want to call it, this became prominent in 2020, okay? Whereas we started to see a little bit in 2019, but especially in 2020, a lot of lilac leaf blight. And it was really alarming for a lot of people because normally lilac is a very hardy, you know, ornamental that you don't really have to worry about. You plant it as a border and it seems to do great. But with this lilac leaf blight, really seems to be the exception to this, and exceptionally widespread in Minnesota. And it was about mid-season, about mid-July, that lilac started to turn brown in that year. In last year, we were thinking, well, you know, it's really hot and dry, so maybe it won't be as much of a problem because it's a fungal pathogen, but we still did see it about mid-August. Okay, so it did seem to take a little bit longer for it to get going, but it didn't actually, you know, start to get going again. So I have every expectation we're going to see this again this year. There was actually an article about it in the Tribune in 2020 because it was so alarming to folks, and one of our diagnosticians was interviewed for that. So again, relatively, it doesn't seem like it's a new pathogen because it has been reported before. So why is it now becoming more of a problem? Is it like a new strain of the pathogen that's more virulent? You know, if we think of like with Omicron and Delta and all these new variants, is it a similar situation going on here? You know, it really does need some research, and I don't know of any group that's getting into this right now, but it's definitely an area that definitely could use some investigation. So as far as overall health effects, you know, because it is kind of a late season disease, it doesn't seem to be hurting the overall health of the tree that much. I know I have a couple of lilacs that have this, and they got really brown towards the end of 2020. Right now, they're looking pretty good, right? And it's a full set of leaves, but it seems to be something that's really affecting the leaves distinctly themselves. And even though it might not have huge effects overall on the health of the tree, because lilacs are an ornamental and it's making the trees really ugly, you know, this can be a problem for people, obviously. So as far as control recommendations go, because it's so new, or at least the significance is new, we don't really have a lot right now. You know, there are fungicides which are labeled for lilac that you could test out, but as far as when is the best time to treat, obviously before they start to get symptoms would be my guess, but yeah, there's still not a lot known about how best to kind of manage this. Okay, so diseases to be on the lookout for, I'll say there's one here at least, and that is boxwood blight. So this is one we've been kind of raising the alarm about, at least in the last 2 or 3 years. And it's a fungal disease, Calanectria pseudoniviculata. It has not yet been found in Minnesota. As you can see on the map, it was first reported in 2011 in the US on the East Coast. It has spread west fairly quickly since that time. Okay, and it is in Wisconsin, but it has not yet been reported in Minnesota. I'm guessing it probably is here, but we just haven't had a positive case of it yet come into the clinic, or, you know, also the Minnesota Department of Ag hasn't yet sort of found it as well. But as you can tell from the name, it's a pathogen of boxwood, which is a kind of an ornamental, usually used as kind of a hedge for a lot of homeowners. And it can cause really severe effects, especially, you know, in some of these areas in the East Coast where boxwood is very prevalent, and, you know, it's really been devastating. And it will be here at some point. It's really just a matter of time. So it's best probably to prepare for that. And the best way of doing that is to plant resistant varieties of boxwood. Okay, so if you are interested in planting boxwood, make sure it's one of the ones that is labeled as having pretty good resistance. And there's multiple, you know, information sources out there right now that are, you know, will give you pretty good information about what varieties seem to be the most resistant. Seems like these American boxwood versus the English boxwoods tend to do a bit better. So definitely don't plant any of the susceptible ones, because my expectation is they're going to get affected at some point. We do have kind of a similar disease called Volatella blight that's in Minnesota that can cause kind of similar symptoms, but much less severe. And the Volatile blight also requires wounds to really get established. Oftentimes kind of winter damage is what it kind of likes to utilize to get going in a plant. Whereas boxwood blight seemingly doesn't need any wounds. That's why it's such a big problem. Okay, so those are all the diseases I want to talk about. Just kind of wrap up here, just kind of give you some quick hints about sample collection. First of all, starting with Oak Wilt lab testing. It's really critical to collect good samples for Oak Wilt because our diagnosis is only going to be as accurate as the quality of the samples we get, okay? In some cases, people will send us branches that have fallen down to the ground and are totally dead. These are very difficult to diagnose because Oak Wilt is not a very good saprophyte. It doesn't compete well with these other fungal organisms that start to get into the dead branches. In other cases, people will send us just some leaves and twigs, which also is very difficult to diagnose from. So really, we need living branches that are at least a 1/2 inch to 1 inch in diameter. So living branches, basically, they still have some green leaf tissue on there in addition to some of maybe the brown tissue, the leaves that they're concerned about. And then also collecting samples during the active wilting season. So what this means is kind of the mid to late part of the summer when oak wilt really starts to express symptoms, that's the best time to collect samples. The reason for this is because if you have oak wilt in a tree, it's not necessarily in every single branch, okay? It can be kind of scattered throughout the tree. And you don't know which branch has it until you actually see the wilting from the leaves. So you could have an oak wilt infected tree, and maybe you wait until the winter to collect samples, and you just happen to collect a branch that doesn't have the fungus in it. And we would have a negative diagnosis at that point. So you might think everything is fine when in fact your tree actually does have oak wilt. So that's why it's really important to collect during the active wilting season. Having said that, during the active wilting season is also when you're concerned about the beetles spreading the disease. So you certainly don't want to be collecting samples from a healthy tree and then have it get infected because you were collecting samples from it because you were cutting branches off. So that's why it's important to immediately coat that cut surface with some sort of latex paint or shellac to prevent, attracting these beetles to that area. Okay, and then after that, it's important to keep the sample cool and submit it relatively soon after collecting. In some cases, we've had samples that have been in the back of pickup trucks for a week or two before they got around to actually bring into the clinic, and they're all dried up and they're very difficult to actually, work with. Okay, as far as general submission recommendations go, we have gone through some changes in the last couple of years. For a while, obviously, with the pandemic, the building actually was closed to the public, like most University of Minnesota buildings were. And so people couldn't get in. So we had a drop box outside that you could drop samples off and give us a call and we'd come and get them. And we still have that drop box for people who are, a little bit concerned about coming into the building, or perhaps because they just want to quickly drop something off and get back to their car that's maybe idling somewhere. So we have the drop box. We're going to keep using it this summer, but the building is open. So if you want to come in and come into the clinic, that's fine as well. Or we have a table outside of our lab that you can leave samples in if the door is locked for some reason, like maybe we're in a meeting or something like that. We definitely recommend living plant material. It can be difficult to diagnose dead plants because other organisms start to come into that tissue. Whole plants are usually best. So, if you have a plant that has leaf symptoms, well, it might be because of a root problem. And if we only get leaves, we're not able to diagnose that root problem. So if possible, it's always good to get a whole plant. And I know in a lot of cases with trees, that's simply not possible without actually damaging the tree too much. So we'll definitely work with what we can get. If you're wondering about what to collect, you can certainly send us pictures and we'll try to guide you about the tissue to collect. You can hand deliver it or you can mail it. If you mail it, we recommend wrapping roots in plastic, maybe the above ground material in newspaper before putting it in a box. What happens in that case is it prevents the plant from drying out because the roots are wrapped up. But because the whole thing isn't wrapped up in plastic, it doesn't get, all mushy like it would if you're just in a plastic bag over the weekend in a really hot week, really hot weather. We have submission forms. It's always good to get as much information as possible about the history of the plant. Are there other plants affected? What was the pattern of the symptoms that you're seeing? Has it been treated with anything? Whatever you can tell us is always useful. And also, as I mentioned before, calling, emailing with questions. We have our email here, our phone number is up here, website pdc.umn.edu. We have our list of fees, more instructions for submitting samples, and other information. But if something is still not sort of questions about, feel free to give us a call. We're open sort of 8:30 to 4:30 throughout the week, mostly Tuesday to Friday, but oftentimes we're here on Monday as well. Okay, that is all I have. And I'm just gonna look at the chat here, see if we get any questions. So Julie had some points about the lilac leaf blight. Yeah, pruning practices. I don't know. That's a good question, whether that would make a difference or not. I have certainly not seen any experimentation on that. And let's see here. Signs or symptoms are required to ID white pine blister rust. So typically with white pine blister rust, it starts in the needle and then it gradually works its way into the branch, and you'll see flagging branches, like you have a white pine with one dead branch, like, and the needles are all dead. And that's kind of a area to look at more closely. And that branch at a certain point will get the pustules from the spores that will kind of encircle it, right? And that causes the branch to die. And then the spores themselves are really what we use to actually diagnose it. Okay, and oftentimes you get a lot of resin production from that area, but that by itself, that can be something else that causes that too. So that's the main thing we look for. I know we're almost out of time here, but any other questions that are out there?
Hey, Brett, I had a question. I'm wondering if you have ever received a sample of bur oak that had oak wilt, two-line chestnut borer, and Bur Oak Blight all in one sample.
I don't recall one. It's certainly possible. There's no reason why you wouldn't have that. We don't typically, that's not the common thing. Usually if someone has an issue with a tree because the leaves are browning, it's oftentimes one or the other that we see. Well, it's either two-line chestnut or it's the oak wilt, right? It's pretty unusual to get both at the same time, but I can't think of any sort of theoretical reason why you wouldn't have that.
And another one, I know you've mentioned the hypothesis before about how it's sort of the precipitation regime that's happening during the leaf emergence or at a specific time for Bur Oak Blight. And maybe this is getting out of your area, but have you heard of other people thinking about trying some sort of preventative fungicide spray, either for Bur Oak Blight or anthracnose on oak?
With Bur Oak Blight, I know some tree care companies have used an injectable fungicide, kind of similar to the Oak Wilt injectable, but I believe they use like a half strength in a lot of cases because bur oaks tend to have, I think they're a bit more prone to the phytotoxicity than some of the others. And I believe, I haven't seen a lot of, real direct data from some of that. And again, it's, it's a little bit controversial because in some cases you can have a tree that seems to be really bad, but then next year it looks pretty good, so I think it's a difficult thing to tease apart, whether controls are effective in some cases or whether not, and again, you're dealing with really large plants, hard to get a lot of, good replicates and design a effective experimental study. So it's kind of a difficult thing to get a handle on.
Definitely. And why don't we end with this one? Did you see the question from TJ here? Are you still finding Larula needle blight in spruce tree branch samples? Yeah,
we do get it sporadically. It definitely doesn't seem to be as common as the stigmina is, but we do see it relatively common. And there was another question I saw here, much canker in your oaks. Botryosphaeria twig canker probably is the most common one we see. And when that happens, you'll get like a clump of dead oak leaves on an otherwise healthy looking branch. Whereas with oak wilt, you know, the leaf itself kind of marginally starts to turn brown, and it seems to be a bit more ubiquitous in the actual branch that you see. So that's kind of a good way of kind of differentiating that just in a, you know, just in a visual inspection. Hypoxylin canker is another one we see in oak, pretty, well, I shouldn't say too uncommonly. And it has a relatively distinctive kind of patch of fungal material that you can oftentimes see on the outside when that happens.
Well, thank you so much, Brett. Really appreciate it. We had a good audience here online, as you see. And thank you everyone again for being here. We'll be back this summer at some point, and we'll hope to see you again then.